In this lecture, we are going to study about enzymes, nomenclature of enzymes, then classification of enzymes. I am Savita More from Arts, Commerce and Science College, Nasik Road. This is 6th chapter from class 11. Enzymes. German chemist Edward Buchner discovered enzymes by accident. Buchner discovered that living cells were not necessary but that yeast extract could bring about fermentation. He then coined the term enzyme. En means in, zyma means yeast. In yeast it is unicellular fungus. This term is now commonly used for all biocatalysts. Thousands of different chemical reactions takes place automatically at a given time in a tiny living cell. The reactions takes place at the body temperature. If these enzymes were not present in the cell, either the reaction would not occur, or if they occur, they would occur at very slow rate. Each enzyme catalyzes a small number of reactions, specifically perhaps only one. The substance upon which an enzyme acts is termed as substrate. The enzymes which act within the cell in which they are synthesized are known as endozymes. Example, enzyme produced in the chloroplast and mitochondria. If they act outside the cell, they are called exozymes. Example, enzymes released by many fungi. So when they are synthesized inside the cell, they are called endozymes. Example, enzymes produced in chloroplast and mitochondria. And if they are produced outside the cell or when they act outside the cell, they are called exozymes. For example, fungi. These enzymes synthesized by living cell retain their catalytic property even when extracted from the cells. Purely proteinaceous enzymes are proteases that split proteins. So ases, whenever it ends with ases, ASES, it refers to the name of that enzyme. Proteases which splits proteins, lipases which splits lipids, amylases which splits amylose. So like this, ASES means it helps to split. Conjugated enzymes are made up of protein to which non-protein prosthetic group is attached. So, prosthetic enzymes are the enzymes. Nature of enzyme which includes purely proteinaceous enzyme like proteases enzyme. Conjugated enzymes, they show the presence of non-protein prosthetic group. Prosthetic group is firmly bound to the protein component by chemical bond and is not removed by hydrolysis. If the prosthetic group is removed, the protein part of enzyme becomes inactive. There are enzymes which require certain organic compounds and inorganic ions for their activity. The organic compounds that are tightly attached to the protein are called coenzymes. Remember, what are coenzymes? Coenzymes are the organic compounds that are tightly attached to the protein. They are called as coenzyme, whereas the inorganic ions which are loosely attached when they are tightly attached to the protein called coenzyme. When they are loosely attached to the protein, they are called cofactors. Some of the organic coenzymes are nicotinamide adenine dinucleotide, that is NAD, and flavin mononucleotide, that is FMN. Inorganic ions of metals which acts as cofactor include magnesium, copper, zinc, iron, manganese, etc. Iron is cofactor of enzyme catalase. Manganese is cofactor of peptidase. Often metal cofactors are referred as enzyme activators. Nomenclature and classification of enzymes. Nomenclature means giving name. Here enzymes are given name on the basis of the substrate on which they act. For example, protease, sucrase, nuclease, lipase, etc., which breaks up proteins, sucrose, lipids, nucleic acid, lipids respectively. The enzymes can be named according to the type of function they perform. Example, dehydrogenase means to remove hydrogen. Carboxylase means to add carbon and oxygen. Decarboxylase means to remove. D means to remove. Remember, whenever the term comes D means to remove or less, etc. Decarboxylase remove carbon dioxide. Oxidases helps in oxidation. Some enzymes are named according to the source from which they are obtained. For example, papain from papaya, bromelain from member of Bromelaceae family, pineapple. So accordingly they are given names according to International Code of Enzyme Nomenclature. The name of each enzyme ends with an ASE and consists of double name. The first name indicates the nature of substrate upon which enzyme act and second name indicates the reaction catalyzed. Example, pyruvic decarboxylase catalyzes the removal of carbon dioxide from the substrate pyruvic acid. Similarly, the enzyme glutamate pyruvate transaminase catalyzes the transfer of transaminase. Trans means transfer. Transfer of amino group. Transaminase means transfer of amino group from substrate glutamate to another substrate pyruvate. That's why the name is given glutamate pyruvate transaminase. Classification of enzymes includes oxidoreductases. These are the enzymes catalyzing oxidation and reduction reactions by the transfer of hydrogen or oxygen. Example, alcohol dehydrogenase. Alcohol plus NAD, that is nicotine amide dinucleotide, in the presence of alcohol and dehydrogenase forms aldehyde and NADH2, that is nicotine amide dinucleotide reduces to form NADH2. Transferases. This enzyme catalyze the transfer of certain groups between two molecules. Example, glucokinase. Glucose plus ATP. In the presence of enzyme glucokinase, it transfer the group, that is from glucose to ATP. Group transfer takes place to form glucose 6 phosphate plus ADP. That is from ATP phosphate group is transferred to glucose. That's why compound form is glucose 6 phosphate and ADP, that is adenosine diphosphate. From triphosphate, diphosphate is formed. Hydrolases. These are the enzymes catalyze hydrolytic reactions. This class includes amylases, proteases, lipases, etc. Example, sucrase. Sucrose, in the presence of enzyme sucrase, forms glucose plus fructose. Lysis. The enzymes are involved in elimination reaction resulting in removal of a group of atoms from substrate molecule to leave double bond. It includes aldolases, decarboxylases and dihydrolysis. Example, fumarate hydratase. Histidine. In the presence of enzyme histidine decarboxylase form histamine plus carbon dioxide. Isomerases. This enzyme catalyze structural rearrangement within a molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as racemases, epimerase, isomerase, mutases. Example, xylose isomerase. Glucose 6-phosphate is isomerized in the presence of enzyme isomerase to form fructose 6-phosphate. Ligases or synthetases, these are the enzymes which catalyzes the covalent linkages of molecule utilizing the energy obtained from hydrolysis of an energy rich compound like ATP, adenosine triphosphate, GTP, guanosine triphosphate. Example: glutathione synthetase, pyruvate carboxylase. Properties of enzyme. Proteinaceous nature. All enzymes are basically made up of proteins. All enzymes are basically made up of protein. Three-dimensional conformation. All enzymes above specific three-dimensional conformation have one or more active sites to which substrate or reactant combines. The point of active site where the substrate joined with enzyme is called substrate binding site. Catalytic property. Enzymes are like inorganic catalysts and influence the speed of biochemical reactions but themselves remain unchanged. After completion of the reaction and release of the product, they remain active to catalyze again. A small quantity of enzyme can catalyze the transformation of very large quantity of the substrate into an end product. For example, sucrase can hydrolyze 1 lakh times of sucrose as compared with its own weight. Specificity of action. The ability of an enzyme to catalyze one specific reaction and essentially no other is perhaps its most significant property. Each enzyme acts upon a specific substrate or specific group of substrate. So, it is termed as specificity of action. Reversibility of action. Enzymes are very sensitive to temperature and pH. Each enzyme exhibits its highest activity at a specific pH called optimum pH. Any increase or decrease in pH cause decline in enzyme activity. Example: enzyme pepsin. This enzyme is secreted in Enzyme pepsin is secreted in stomach. Shows highest activity at an optimum pH of 2 acidic. Trypsin in duodenum is most active at an optimum pH at 9.5, which is alkaline. Both these enzymes like pepsin and trypsin are protein digesting enzyme. So remember, protein digesting enzyme pepsin and trypsin. Pepsin is released in stomach and optimum pH of pepsin is 2, which is acidic, and trypsin, it is pH is 9.5, which is alkaline. Temperature. Enzymes are destroyed at high temperature of 60 to 70 degrees Celsius or below. They are not destroyed but become inactive. This inactive state is temporary and enzyme becomes active at suitable temperature. So remember this, when temperature is increased more, that is 60 to 70 degree, or decreased less, enzyme becomes inactive. When suitable temperature comes, enzymes become active. Most of the enzymes work at an optimum temperature between 20 degrees Celsius and 35 degrees Celsius.